Hello children, in the last lecture we have studied the details of light and dark reaction. Now today we are going to take what are the factors which affect photosynthesis? Factors affecting photosynthesis. There are various factors. The first factor which affects photosynthesis is light. It is the most important factor. Normally plants utilize sunlight, but marine algae can perform photosynthesis even in moonlight. If it is a case of marine algae, it can photosynthesize even in moonlight. Plants can also perform photosynthesis sometimes in artificial light also. Highest rate of photosynthesis. Highest rate of photosynthesis is in case of a red light, and minimum minimum rate is in green light, as I already told you children. Now actually very high light intensity can cause reduction in the rate of photosynthesis by causing denaturation of the chlorophyll molecule. The second important factor after light is temperature. Now optimum range for temperature for photosynthesis is 25 degrees Celsius to 30 degrees Celsius. In some forms like algae of hot spring. In fact, hot spring algae can photosynthesize even in higher temperatures, that is around 60 degrees centigrade to 70 degrees centigrade, which is normal for them. The next important factor which affects photosynthesis is carbon dioxide. It is the first limiting factor. So, around 0.032 to 0.1% is present in the atmosphere. So it is a limiting factor in photosynthesis. More carbon dioxide is there, more photosynthesis will be there. Now, next factor is oxygen, that is O2. Higher concentration of O2 stops photosynthesis. High concentration, it stops O2, or we can say oxygen is inversely proportional to rate of photosynthesis. Now, the last limiting factor which affects photosynthesis is chlorophyll. Chlorophyll content is directly proportional to rate of photosynthesis. That is, more chlorophyll, more will be the rate of photosynthesis. No photosynthesis occurs in etiolated cells. In variegated leaves, it occurs only at places where chlorophyll is present. Now, significance of photosynthesis. What is the importance or significance of photosynthesis? The first and foremost importance is it is required for the production of food material. Production of food material. And second importance is it helps in the atmospheric control and purification of air. Now, this was all about the factors affecting photosynthesis as well as the significance and importance of photosynthesis. Now, we'll try to find out some of the activities which are related to photosynthesis experiment. The first activity, it will denote an experiment to demonstrate that chlorophyll is necessary for photosynthesis. Now for this, what we will do, we will first place a potted plant, potted croton plant or plant with variegated leaves in dark for 24 hours to make leaves starch free. We'll first make leaves starch free. Then after 24 hours, we'll put this plant in sunlight for a few hours and pluck some leaves from the plant. Now in the next step, we'll boil these plucked leaves, boil the leaves in water for a few minutes, and then we'll boil these leaves in alcohol. First we'll boil it in water and then in alcohol till they become colorless. Now we'll remove the leaves from alcohol, wash and place it in a petri dish. Remove the leaves from alcohol and we'll just put it in a petri dish. Now we'll add a few drops of iodine solution on one of the leaves. Some of the portion which was green in the alive leaf has turned blue-black. Now due to this iodine solution which was green, this will turn that portion of the leaf into blue-black color. This shows the presence of starch in this region. The remaining portion does not develop any color other than that of the iodine solution. So this experiment clearly indicates that green portion of the croton leaf manufactured starch during photosynthesis when placed in sunlight, but the remaining portion which was not green did not manufacture starch, hence chlorophyll is necessary for photosynthesis. We can show the leaf like this. Suppose this was the leaf. It consists of some of the green portion and some non-green portion. After the experiment, this leaf, this will become blue-black in color, which was green. The corner portion which was green, due to the iodine solution, it will be converted into blue-black color, and the portion which was non-green, it will look like that only, showing no color change, which shows that chlorophyll is necessary for photosynthesis. Now there is one more activity to show that carbon dioxide is necessary for photosynthesis. Now let me show you an experimental setup by this. Let me draw a diagram. Here I am showing the sun with beautiful sun rays. The sun is shining brightly. Now there is a potted plant. This potted plant is showing some green leaves. Let me draw this diagram. There is an experimental setup which is like this. This bottle KOH and this is a wide mouth bottle. This is a croton plant, and here this I am showing a leaf with starch and no starch. So starch, and this is no starch region. Now this is an experimental setup. Now what we'll do, we'll select a potted plant having long leaves, de-starch its leaves by keeping the plant for 2 or 3 days in a dark place. Now we'll insert half of the leaf in the bottle through a split cork as it is shown here. There is a split cork over here. Some concentrated potassium hydroxide solution has already been kept in this bottle. KOH will absorb all the carbon dioxide present in the bottle. Now place the apparatus in sunlight. Test the leaf for starch after 3 to 4 hours with iodine solution. Only that part of the leaf will show blue color which was present outside the bottle and therefore received carbon dioxide. Now this experimental setup is also called as a mole's half leaf experiment to show that carbon dioxide is necessary for photosynthesis. Now there is one more experiment, that is experimental setup. This experiment will demonstrate that light is essential for photosynthesis. Light is essential for photosynthesis. In this, what we can do, we will take a potted plant and de-starch its leaves. First, we'll take potted plant and de-starch the leaves of a potted plant by keeping it in dark
for 2 to 3 days.